Okay, so welcome everyone to this session about geometry and structure of the human brain and manipulating those geometries and structures. Noah Benson is a data scientist at the eScience Institute, so he's my colleague at the University of Washington. He joined the University of Washington quite recently, or maybe it's been a little while. He joined here in March. Incredibly enough, we haven't yet met face-to-face since he joined the University of Washington, moved here to Seattle from New York. He was at NYU before as a research scientist. And he'll be presenting this presentation and tutorial. Take it away.
All right. Hi, everyone. Thanks for coming to my tutorial on brain structure and geometry. Like Ariel said, I'm Noah Benson, and I'm a senior data scientist at University of Washington's eScience Institute. I mostly study visual neuroscience, but I'm especially interested in the brain structure and how that structure is related to the brain's function. So what you're seeing right now on the right is a video of a right hemisphere that's morphing from one shape into another. I actually made this video using subjects from the Human Connectome project. And now to some extent, this is just a fun, fleshy video, but let's think for just a minute about what it takes to make a video like this. First of all, we have to be able to collect structural MRI images of a bunch of different brains. Then we have to process them to figure out where the brain is. We have to find the surfaces and model them by using some kind of 3D model of the brain. And finally, if we want to be able to morph one model of a brain into a model of another brain, we need to move each point on the first brain's surface into some kind of nearest point on the second brain. And this requires some kind of information about anatomical correspondence between the brains. So really, this animation is demonstrating some very sophisticated processing, and we're going to be walking through much of that processing today. Now, we're not going to end by making this movie, but we're going to end by at least having gone through most of the tools you would need to make it. Okay, the plan for today is that we're going to go over 4 main topic areas. I'll start by giving a quick introduction to the kinds of measurements we'll be using, primarily MRI. Next, I'll talk in more detail about 3D images, which are also called volumes or volumetric images, and how they can be used to store brain data. After that, we'll move on to the cerebral cortex and the cortical surface, including methods of interpolating between volumes and surfaces. And finally, we'll talk about methods of aligning and comparing subjects' brains. After each of these last 3 sections, we'll be flipping over to the tutorial notebook to go through some demos as well. So right now, I'm going to go over to the tutorial notebook, which I have here. This is running on the hub. So if you want to open this on the hub, you can just go back to the home, go to curriculum, go to somewhere in here is Wednesday, geometry, Benson, and then you can open up the class notebook here. And just to get us started, I want to mention that this notebook is intended as a reference for you to come back to later. There's a lot of text and a lot of comments in this notebook that I'm not going to go over because they're either intended to show alternate ways of doing things, or they're intended to help you if you return to this later to understand things without the context of me talking about it. So just so you know, this will be easy to come back to later. It's all set up with a Docker compose file that you can see here, so that even after the Jupyter Hub is shut down, you'll be able to run all this notebook inside of a Docker. And if you have questions about that, I'm happy to answer it after the talk. But for now, let's start by just initializing our notebook over here. I'm going to start by importing some standard libraries, including NumPy and SciPy and Matplotlib for 2D plotting. I'm also going to import a library called IPyVolume, which does 3D plotting. And then NeuroHackademy, which you're probably familiar with. There's a tutorial about that earlier today. And then finally, I'm going to import a library called NeuroPython. This is a library that I maintain that's going to be the main library we can look at in this notebook. And it builds on top of NeuroHackademy to provide sort of structure around the various files. And a lot of the structure is built around representing subjects as opposed to individual files. And hopefully that will become clear as we go through this notebook. So I'm just going to go ahead and evaluate these 2 cells. And then I'm going to go back over to my slides, and we'll come back to that notebook throughout this talk. Okay, so for starters, I'm going to give a quick kind of introduction or review for magnetic resonance imaging. So my expectation is that many of you have already worked with MRI, and for you, this may be a bit of a review. For some of you, though, you may only associate MRI with the kind of machine shown on the left here, and with the kinds of output images shown on the right, which might at first glance look a little like CT scans. In fact, MRI stands for magnetic resonance imaging and is a method of imaging chemical properties such as the density of a particular molecular structure within some space. So it's very different than imaging methods like CT. Now, there's a lot of science and complexity behind how MRI machines work, and I'm not going to discuss that today, in part because I'm not really qualified to teach MR physics, but suffice to say that a big part of MRI work can be determining which scan parameters to use for an experiment. However, that's going to be out of scope for today. The images on the right are actually all slices of different kinds of MR images that can be produced by a single MRI machine using different scan parameters. These three images represent three of the most common kinds of MR images that are used in neuroscience. On the left is a T1 weighted images, sometimes just called T1 or an anatomical image. And in a T1 weighted image, the fat typically shows up as brighter, while water tends to be darker due to the scan parameters. T1 weighted images are incredibly valuable images for determining the structure of the brain, and that should be kind of obvious just from looking at the image. In the middle is what we call a T2 weighted image, and T2 weighted images are a bit like T1 weighted images, except that the fat appears dark while water appears bright. So they're not quite as useful for T1 as T1 weighted images for structure, but for example, you can combine T1 and T2 images to estimate myelinations. So they do show up quite a bit. The last image type is a bold image, which stands for blood oxygenation level dependent. In bold images, oxygenated blood is light, and deoxygenated blood is dark. So you can use these images to infer neural metabolism, which can be used to infer neural function. Bold images tend to be taken very quickly, so that you can take many images during, like, over the course of a single scan or experiment. And partly because of this, they tend to have much larger voxels and lower resolution than the images on the left. And notice that these two images on the left give a lot of detail about anatomical structure, and for this reason, sometimes we call them anatomical or structural images, while the image on the left is what's known as a functional image. Now, we won't be discussing functional measurements very much more than we already have today, but I wanted to start by pointing out that functional measurements are greatly enhanced when accompanied by structural measurements, and this is true for many modalities, not just MRI. Here are a few examples. So obviously with functional MRI, we tend to need T1 weighted images to know where the brain is. For electrocorticography, which is also sometimes called intracranial EEG, it is usually necessary to align a structural MR image with a CT scan of the implanted electrodes in order to deduce the location of electrodes in the brain. Similarly, for EEG and MEG measurements, source localization generally requires a T1 weighted structural image. And just in general, for almost all neuroscience work, visualization of brain data is often best when performed on a 3D model of the surface, and the best methods of inter-subject comparison tend to require high-quality structural data. So for all these reasons, our best friend in studying brain structures is the T1 weighted image. T1 weighted images are very high resolution. In 3T scanners, you can usually get voxels that are about 1 cubic millimeter in size, although they can be even smaller with 7T scanners. T1 weighted images can be collected very quickly, usually just a few minutes, and they, as you can see, they give us very good data about brain structure. In fact, if we zoom in on just a little patch of the brain here, we should be able to it should be pretty obvious that you can make out the skull and the brain in a little space between. Additionally, we can see a few sulci in this image here that are pretty clear. And around the sulci are darker, slightly darker voxels, while further away are lighter voxels. These lighter voxels constitute the brain's white matter, and the darker voxels around the sulci represent the brain's gray matter. Now, just as a quick review, cortex is actually composed of two kinds of tissue, gray matter and white matter, which I've highlighted in this image here. And noticeably, you can also see the cerebellum and brainstem in this MR image, but I'll be focusing quite a bit on cortex during this talk. The white matter makes up an inner core of the cortex, with the gray matter forming a thin layer on top of it, as shown in the upper right corner, or on the right here. And just to explain cortex really quickly, I'm going to make an analogy to a computer cluster. So the gray matter of cortex contains most of the neural cell bodies. Thus, we kind of think of it like the CPUs or compute nodes of the brain, while white matter, on the other hand, is mostly made of neural axons, which are sort of like long-range connections between distant parts of cortex. In this way, the white matter is more like the wires connecting computer nodes. Okay, so that was just a quick introduction to structural MRI. We're not going to go over the notebook just yet. But for now, let's go ahead and dig a bit into the details of how we represent and interact with 3D image volumes. So what you're seeing here is an actual T1 weighted image that is just rotating. So clearly this image is not only a 3D volume, but is actually a pretty detailed one. You can kind of see the level of detail here. So I want to draw your attention to just a few things about this image. First, as you can probably see, there's something a little bit weird about this subject's mouth. That is actually because this subject has a non-ferrous metal retainer underneath their bottom teeth, and that causes just enough distortion of the magnetic field that the entire mouth gets kind of blurred out. You might also notice that it looks like the subject's ears are kind of matted down to the head. This is because, generally speaking, we want the head to be very still during a scan, so we tend to put a bunch of padding around the head when we scan people. In fact, if you're very observant, you might notice that this is knee. And in fact, usually if you look at a 3D image like this, the face will be cut off, and that's before anonymity. But I wanted to show you guys a face, so I used an image of myself, which allows me to not anonymize it. So when viewing a raw image like this, the image looks a bit like a laser scan. However, there's a lot more detail here than you're actually seeing right now. And to show that, if we just clear half of the image, which you should be seeing now, you can see that there's a lot of data here. It's not just a superficial scan. So when people talk about volumetric data or volumes, they're talking about 3D images like this. And of course, we're going to be focusing on what we're going to be focusing on for the rest of this talk is specifically the brain, which you can see here. And however, my ability to show you this subpart of the image that is just the brain kind of begs the question. Given a T1 weighted image, how do we know which voxels in the MR image are part of the brain? So the question of which voxels correspond to what anatomical features is one of the first major tasks of anatomical image processing. This job is called segmentation usually, and it used to be long ago, used to be performed by hand. Researchers would actually go and look at every slice of their image and click on every voxel that was part of the gray or white matter or some other structure like the cerebellum. These days, there are a number of programs that will do this for you, so you don't usually have to do it by hand, although you often will correct these by hand. These are some tools that can auto-segment T1 weighted images. I'm going to be, during this talk, mostly focusing on FreeSurfer. And part of the reason for that is because FreeSurfer is one of the oldest and most commonly used of these tools. Additionally, the HCP pipelines or human connectome pipelines, which were used as part of the Human Connectome project, they are basically, they run FreeSurfer and then perform additional analysis. So FreeSurfer and the HCP pipelines are very similar. And VistaSoft, the last one listed here, is just a MATLAB-based example, and it can work with FreeSurfer as well. So FreeSurfer is actually a very pervasive kind of tool. So we'll come back to FreeSurfer in a moment, but for now, regardless of which preprocessing tool you pick, the results of the preprocessing is going to be a bunch of 3D image files that contain various kinds of data and labels. And in order to understand these, we need to first understand a bit more about how 3D image data are stored. Now, I think that you guys have already gone over a lot of this in the NeuroHackademy tutorial, so these next few slides I'm going to breeze through just a bit. Basically, MRI volume files or 3D image files, there's a bunch of different formats, and they all contain different kinds of data, but they almost all of them contain, at the very least, a 3D or 4D array of voxels. If it's 4D, it's because it's something like a bold image where you've got the fourth dimension as time. And additionally, they almost always contain an affine transformation matrix for alignment of the volume with some external coordinate system. A lot of them also contain additional metadata, such as scan parameters or units like millimeters and seconds. And there's a variety of file types. Nifty is the de facto standard. FreeSurfer has its own format, which is very similar to Nifty. Scanners usually produce DICOM files. Usually the affine transformation, I believe you guys actually talked about affine transformation matrices quite a bit in the NeuroHackademy tutorial. So again, I'm going to go through this very quickly, but affine transformations are usually represented as 4x4 matrices, kind of like shown here. Just very quickly, the idea is that usually you would take a set of coordinates, x, y, z, or row column slice for a volume, and multiply it by this matrix and get some new matrix that comes out some new coordinates on the other side. And this affine transformation matrix generally stores things like rotation, resizing, and reflection in this upper left corner, and translation in this sort of column vector here. And it can hold any linear transform basically. In addition to rotation, resizing, reflection, you can get things like shearing as well. But for neuroscience, these generally are the three kinds of transformations we care about. And yeah, like I said, this usually operates over row column slice of the image or of a particular voxel. So you sort of take each voxel, multiply it by the transformation matrix to get the new coordinates. I think I will skip over some of this. You can always return to this later as reference, but I think this was covered in the previous lecture, so I'm going to go through some of this kind of fast. Additionally, there's orientation information that is sometimes stored in the voxels, and mostly I just want to draw your attention to the fact that when you're looking at 3D images, it's often not intuitive how the image is oriented. So if you look at this image on the right, over here, you can see, you might kind of naturally assume that this side of the right side of the image is the right side of the brain, and the left side of the image is the left side of the brain. And that would be true if you were looking at the brain from the perspective of a neurosurgeon. But if you're looking at the brain from the perspective of a radiologist, it's actually flipped, because you would be looking kind of up through the brain instead of down on top of the brain. So without going into a whole lot of detail here, I just want to make this warning that if you receive MRImage files from someone else, or even your own scanner, make sure you know which side is right, which side is left, because it's not always obvious. Okay. So fortunately, a lot of the complexity of processing MR images that I just kind of breezed past is handled by the various software that I mentioned earlier. So FreeSurfer is one of the most common examples of these programs. FreeSurfer performs a whole bunch of anatomical processing steps that we will discuss throughout this tutorial, starting with volumetric image processing. We'll talk a bit about FreeSurfer as an example of this process, though other programs will perform similar operations. So this slide I've partly included as a reference that you can return to about FreeSurfer in the future. When you run FreeSurfer using its main pipeline interface, ReconNull, which by the way is a real pipeline, it runs many, many programs. It organizes your data into a directory for your subject inside of a superdirectory where it keeps all the FreeSurfer subjects called the subject stir. And this is typically stored in an environment variable with the same name. For more information on all these steps and how they work, you can visit the various FreeSurfer websites listed here, and they'll be on the final slide as well for reference. Now, if we take a look at a typical subjects directory, as shown in this terminal screenshot, I've just basically changed directory to the subject stir and done an ls command. We can see that it contains a number of subdirectories. Now, all of these directories here with the name fsaverage or fsaverage4, etc., these are part of an anatomical template that we'll be talking about later in the last section of this talk. These directories called, for example, BERT and S1202, they're just regular subjects that I've scanned. BERT actually is a built-in subject that's an example subject for FreeSurfer, but S1202 is a subject that I scanned. The other subjects, for example, V1_average and CSV_AVG35 are built-in FreeSurfer data that I'm not going to talk about in this talk, but it's pretty normal to see them. So if we then look inside one of the subjects directory, BERT in this case, we see another set of subdirectories. These directories contain the various subject data, including MR images and the processed MR images, various surface models, and a variety of metadata such as Brodmann area labels. So I won't go over too much more detail of the FreeSurfer subject directory than that, but at this point, let's go back over to our notebooks to look at some of these data a little more closely. Okay. So the subject that I showed you the rotating 3D image of, if you want to look at that subject, it is subject NBen, but you can also use subject BERT if you would like here. What we're going to do is we're just going to ask NeuroPython, that library we imported, for the FreeSurfer subject object for that subject ID. Go ahead and run that, and let's see what it gives us back. You can see that it's given us back an object that's some kind of subject object. It seems to know where the subject is stored. Okay, so let's go ahead now and load an MRImage file. So there's a few ways to do this. I'm just going to quickly go over one of them. Here we can just ask the subject to load a file from its subject directory. You could also do this just plain with NeBabel. But when we do that, you see that we get back a NeBabel image. So clearly, this is just sort of a wrapper around NeBabel in this particular case. And just to prove that it's the same, we can go ahead and use NeBabel to actually load that exact same file and then print the file names. And we see that they're both the same. So we can load it that way. The The subject object also keeps track of various images, and based on its position in the FreeSurfer processing pipeline, T1.mgz is not actually the original T1, it's an intensity normalized T1. So NeuroPythy's subject object can give you back that same that same image, and this is just a test that they're the same by checking that all the voxels are the same. And if we run that, we find that they are. Okay, so these are just a few ways to read in some image files, but now let's go ahead and visualize some of them. So I have a code block here, and I realize that in the NeBabel tutorial, you probably looked at some of the like orthoview methods and stuff. This is just a manual way of doing approximately the same thing. So what I want to do is I want to plot 3 different slices out of this image. And I'm going to take the slices along axis 2. This can be either 0 for the rows, 1 for the columns, or 2 for the slices. And I'm just going to take these 3 slices. I've just kind of picked these out of the middle of the image. Now what I'm going to do is with matplotlib, I'm going to set up a figure that has 3 panels. And then I'm going to iterate through the slices I picked out and just plot based on whichever axis I picked. I'm going to pull out, in this case, here I'm going to pull out the slice that I selected and just plot it with ImShow, matplotlib's ImShow. And that should give us 3 slices of what looks like a T1. Okay, so this is nice, but this is nothing but a rotating 3D image that I showed you earlier. So I wanted to show you that you can actually look at 3D images like this in a notebook like this. So I'm going to use IPyVolume, that 3D plotting library I mentioned. We're going to make a figure, and then we're going to call this quick vol show function on the actual array stored inside of that image. And then I'm going to tell IPyVolume to plot the image. Now this will take a minute, and it may even take a little longer on your notebooks. And additionally, it will produce this error, which can be ignored. But in just probably about 20-30 seconds, we should see a 3D image pop up here. I see this is maybe a good time for me to look at questions. Can I speak about the nuanced differences between choices of pre-processing software, pros and cons? That's actually a really big topic. So I don't know that I can quickly answer that question. However, I'd be very happy to discuss it after at some point on Slack after the talk. In general, I use FreeSurfer the most because it's one of the best ones for cortical surface processing. HCP pipelines is very similar. So my general recommendation would be to use one of those, but we can talk more about that here. Okay, so it looks like this is plotted, and this is actually an interactive 3D plot, as you can see. There's a whole bunch of little widgets you can interact with that will change the coloring and such. I mostly just wanted to show you that this was possible, so we won't mess around with that very much right now. Okay, so I mentioned that FreeSurfer does a whole bunch of what's called segmentation, where it picks out various anatomical segments of the images. So I'm going to go ahead and look at that next. I'm going to ask for the segmentation image of our particular subject. There's a couple other ways to get that same image in the comments there. And then I'm going to use that exact same bit of code I used a minute ago to plot the 3 slices. It's the exact same piece of code. I'm just using the aseg image that I just grabbed, the segmentation image, instead of the T1 image. So if we plot that, what we can see pretty clearly is that we get something that looks like a brain that comes out the other side, but the grayscale values are a little unclear what they mean here. And that's because this segmentation file is actually full of labels. Every voxel has an integer label value, which corresponds to this, the particular piece of anatomy that it represents. So to understand which number corresponds to which segment, we can look at FreeSurfer's, what's called FreeSurfer's color LUT. I don't actually remember offhand what LUT stands for, but it's basically just a file that contains, for every label, a name and a color that FreeSurfer likes to use to plot that particular label. You can load it like this using NeuroPythy. And what you get back is just a pandas data table where you have a label number and then the name of the label and then a color. This is an RGBA value. So as an example, let's suppose we wanted to just look at the part of these images that was left, the left gray matter. So we would go in here and we find number 3 is left cerebral cortex. So we know that any voxel with a value of 3 in this particular image will be left cerebral cortex. So I'm going to say our label is 3, and then I'm going to rerun that exact same block of code that I ran earlier, except I'm going to add a little line here that says, basically, instead of keeping the segment slice, I'm going to convert it into a Boolean mask where it's true anywhere the label is 3 and false everywhere else. And if we plot that, what we get back is something that looks a lot like gray matter of Cortex. And this tells us that actually this side of the image, the bottom side here, is the left side. Okay, and then finally here, I just wanted to plot to show you that you could take this color LUT data and replace the actual labels with the colors to get an actual nice colored image of these segments. That's just done in these couple lines here where we basically make an RGBA image, and for every label we just insert the colors wherever that label occurs. And when we plot that, we see these nice all the various segments that FreeSurfer recognizes in these slices. Okay, so that's a quick kind of demo for volumes. At this point, let's go back to the slides. Okay, so we've taken a look at volumetric image data and how it's stored and manipulated, and we'll now look at models of the cortical surface. So clearly, if we look closely at a slice of a T1 weighted image, we can see both what is called the white surface here in green, and that's the boundary between the brain's white matter and gray matter, as well as the peel surface, which is the boundary between the gray matter and the outside of the brain, and that's in yellow here. We can use the difference in intensity between the layers of the image to come up with a model of where these surfaces are in this slice. And if we do that for all the slices, we can come up with a model of where the surface is everywhere, which looks like these sort of surface models that you've probably seen before. The models of the cortical surface are actually triangle meshes, which we can see if we zoom in very close. Triangle meshes are how most 3D models are represented in other fields, including, for example, industrial design and 3D gaming. In theory, this means there's a lot of software already made for manipulating these models. And while this is partly true, many of the problems that occur in neuroscience are not common in areas like 3D gaming, unfortunately. As a general rule, there are a few things that are unintuitive and kind of universally confusing about how surface meshes are usually represented and stored, both in files and in pretty much any software you use. First, the triangles are always stored or represented in a counterclockwise order, such that in triangle ABC, like the triangle you're seeing here, the vertices A, B, and C are in a counterclockwise order around the outward-facing surface normal of the triangle. So this means that if you want to know which side of a triangle is outside of the brain, you just need to calculate vector AB cross vector AC, and that will give you a normal vector pointing out of the brain. Additionally, and somewhat unintuitively, there is no coherent ordering at all to the vertices or the triangles of the cortical surface, or at least you should not expect there to be. A lot of people start out by assuming that the vertices are going to be in some kind of sorted order, but they're not. For example, the vertex with the lowest y value will be the very first vertex or something like that. That is not the case. The first and second vertices are as likely to be on opposite sides of the mesh as they are on the same side. And additionally, there's no correspondence between the ordering of the vertices in one subject and another, or for that matter, in one hemisphere and another. To be a little more specific, the vertex ordering of a hemisphere is very important. Although the left and right hemispheres of a subject will have a different set of vertices, all of the surfaces of one hemisphere will have the same number of vertices, and those vertices will be equivalent. Specifically, this means that you can think of the vertices on the peel surface as being the same as the vertices on the white surface, just displaced outward towards the peel. Vertices are a little bit like lines drawn through the cortical gray matter, and that's kind of what this figure is supposed to represent on the left. Now, this means that when we look at the most commonly used surfaces of the brain, which are shown here as the white, the mid-gray, the peel, the inflated, and the spherical surfaces, the set of vertices in each is identical. It's just been displaced or warped to make a different shape. So each of these each of these meshes is just a distorted version of the others. The inflated surface is made by just smoothing the surface until it's relatively flat, and you've probably seen it before because it's used very commonly for display purposes. And the spherical surface is also made by flattening and inflation, but we'll talk more about that in the last section of the talk. The overall shape of the cortical surface is one aspect of cortical structure, but we want to be able to track additional data on the cortical surface as well. So one example of this kind of data is the curvature. On the previous slide, we've been seeing binarized curvature maps plotted as kind of an underlay. Another example of structural data is the gray matter thickness, and this in particular is very difficult to calculate without surface mesh models. It's very difficult to calculate this from a volume, and yet it's a very interesting measure for a lot of studies. Yet another example would be anatomical labels, kind of like what we saw in FreeSurfer segmentation data, but you can get different and better labels for the cortical surface using intersubject alignment, which we'll talk a little more about later. When we're storing these kinds of data, the data are typically stored just as a vector, with a value for each vertex. So in a curvature file, you would just simply find a long vector of curvature values in the same ordering as the vertices on the cortical surface. In general, the files used for surface knowledge representations are much simpler than people assume. So kind of like I just said, surface geometry files, that is, almost always contain two matrices. One is for the vertices and one is for the triangles. So the triangle matrix just gives integer indices into the vertex matrix for each of the triangle's corners. Property files store one value per vertex in the same order as the surface files, kind of like I just mentioned. In fact, this is such a simple task that one of the more common file formats used with surface data like thickness is the MGZ volume files used by FreeSurfer. And what people do is they just put a 1 by 1 by n volume, where n is however many vertices you have. FreeSurfer also has some curve files that are made for these kinds of properties, but they're not commonly used outside of FreeSurfer, so I'm not going to talk about them. And you may have touched on GIFTI and SIFTI files from the Human Connectome project. GIFTI files are XML based and can store all kinds of data. They're a little too broad for me to go into in this talk as well, but they're another surface data format. Now, suppose we've modeled our suppose we've modeled our surfaces from our structural MRI volumes, but we also collected some bold functional data. We likely want to be able to project that functional data, or some result of analyzing those functional data, like a GLM coefficient, for example, onto the cortical surface for further analysis and for visualization. So this is actually one of the most common questions I get over email, is people asking me how to get data from a volume onto the surface. So step one is basically to find the affine transformation that aligns the volume with the surface. This can depend a little on the software you're using, and usually it will be somewhat encapsulated in whatever method you're using for interpolation. But this is basically the first step. And for things like FreeSurfer, you usually need to align the image with the subject's brain.mgz file, which is one of the files that FreeSurfer produces. Step two is then to invert this transformation so that you can apply it to the surface vertices. Basically, you want to take the surface vertices and you want to transform them such that the XYZ is now in row, column, slice space. So we can then apply that inverse transformation to get those row, column, slice positions, and then we can do some form of interpolation. Now, to understand how linear interpolation works in the volume, imagine that we have a surface vertex, which is shown here in black, in the very middle of this kind of cube. We imagine that it falls somewhere in the voxel space, as shown here, and the centers of the 8 nearest voxels to the vertex are shown as colored and grayscale dots all around it. In linear interpolation, we want to assign the surface vertex the value that is linearly continuous between all the voxel centers for that vertex position. In nearest interpolation, obviously, you would just take the closest vertex, which in this case is this light gray vertex, and assign the surface vertex that value. But for linear interpolation, we want to assign something continuous. And there's a pretty simple, or at least relatively intuitive formula for how to do this that is kind of displayed here. The idea is that we want to give each of the 8 vertices, or excuse me, voxel centers around the surface vertex a weight that is dependent on how close the particular vertex, the surface vertex, is to that voxel. So what we do is we split this cube into 8 subcubes, and we take the particular subcube that is opposite the vertex in question. So the weight for this light gray vertex is equal to the relative volume of this light gray cube. Relative volume being the total area of this cube divided by the area of the whole cube. So for the light gray, we match this very close light gray voxel up with this very large light gray volume, whereas for this dark gray voxel center, we it up with a much smaller cube back here because it's much farther away from the surface vortex. We basically take the dot product of the values in each of these voxel centers with the weight, and that will give us an interpolated value. Now, we can also interpolate in the other direction from the surface, from the surface into the volume. But this is quite a bit trickier and out of scope for this particular tutorial. But we will we'll touch on it a little bit later. For now, we'll go to the notebook and look at some examples of surface data as well as how to perform both kinds of interpolation using NeuroHackademy. Okay, so here we are back in the notebook. The first thing we're going to do is we're just going to load in a surface geometry file. And we're just going to load this using eBabel so we can see what it actually contains. So feel free to change these if you like. If you want to make, you want to do the right hemisphere or you want to do the peel surface instead of the white surface. Just don't pick the mid-gray because the mid-gray isn't actually a FreeSurfer surface. It's one that we calculate from the peel and the white. I'm just going to go ahead and use the left of the hemisphere of the white surface for now. And I'm just going to grab the path for this particular file. It's actually just the subject's path /surf/lh.white in this case. For peel, it would be lh.peel. So we'll get that file name, and then we're just going to call MRIbabel's read geometry function to read in that file name and just see what it returns. So what it's returned here is a 2-tuple with 2 numpy arrays in it. The first looks like an array of coordinates, it's like n/3, and the second looks like an array of triangle indices, m/3. So basically, it just contains exactly the data that we talked about. Now, it happens that because this is such a common format for these kinds of 3D mesh models, that library IPyVolume that we looked at earlier has a built-in function just for plotting these. So I'm going to go ahead and grab the coordinate matrix and the triangle face matrix out of that surface data tuple. I'm going to grab x, y, and z out of the coordinates, and then I'm just going to make a figure and call that function just like this, x, y, z, and then triangles equals faces. I'm going to set the x limits to be pretty equal, and then go ahead and show the plot. And that will take just a second. But here we have a plot of that cortical surface, and that looks like white matter to me, although obviously the grains are not red, but we'll deal with that in a minute. So as useful as it is to be able to plot something like this, something like the cortical surface like this, we really like some tools that make this a little bit easier and that kind of track the fact that the white and the peel and all these other surfaces are all kind of related to each other. So what NeuroHackademy does is it has what's called a Cortex object that represents an entire hemisphere and tracks all the surfaces of it. So what I'm going to do here is go ahead and grab a Cortex object. You could actually replace this by Cortex equals subject.lh or subject.rh, but since you might have picked RH or LH, I'm doing it this way. This is just an alias. And let's see what that Cortex is. So what this gives us is a Cortex object. It knows its left hemisphere, has a bunch of faces and a bunch of vertices. So that seems pretty normal. Now, the Cortex object tracks a bunch of different surfaces, as I mentioned, the white, the peel, etc. So let's just take a look at which surfaces it tracks. This is just a dictionary. We can see here that it has a representation of the peel, the mid-gray, the sphere, the inflated, the white, and then 2 variants on the white surface. The first is the white original, which is sort of like the very first original mesh FreeSurfer comes up with before any post-processing. And then the white smooth, which is a highly processed smooth mesh of the white matter surface. And if we look at one of these surfaces, what we get back is a mesh object, which again has a certain number of faces and a certain number of vertices. Okay. Now I just wanted to prove to you that the white and the peel surfaces both have the same face matrices. So here I'm just going to check that the triangle face matrices are equal for both of these. And the answer is true. So that's intuitive. And I also wanted to point out that these mesh objects uniquely carry certain geometric information. For example, the area in millimeters squared of each of the triangle faces, or the distance between neighboring vertices called edges. You can also get the list of edges here, so let's just look at these. Yeah, so each of these is just a vector of a single value per in this case per triangle. So this is the very first triangle, has an area of 0.14 millimeters squared. And here we can see that the very first edge has a length of about 0.5 millimeter, etc. Okay, so now let's talk a little bit about vertex properties. Properties are something that are tracked by the Cortex object. So if we want to look at properties, we can just ask for this dictionary of properties. Here I'm going to look at all the keys. Okay, so there's quite a few properties that are tracked by NeuroPython that come from FreeSurfer. Some of these, for example, thickness down here, or surface area, for example, or curvature, these are just the kind of properties we talked about. They are just a value per vertex, and they carry some kind of morphological or geometric information. Additionally, you have a bunch of properties that come in pairs that are, for example, label and weight. So a label is basically just an ROI. This is a property that is true wherever there's a vertex in the label with ROI, and false where it's not. The weight, on the other hand, is pretty much equivalent. It's just that it gives you a probability for every vertex as to being in the ROI or not the ROI or not in the ROI. So a high value of the weight indicates it's likely to be in the ROI. And these are made by looking at intersubject comparisons. So we'll kind of return to this a bit in the next section. Now, something that's worth noting is that not every version of FreeSurfer produces the same set of properties, and not everyone distributes all of the properties when they distribute their subjects. So it's pretty common that some of these will not actually be part of a particular subject. And in this particular case, the subject that we have doesn't actually have any of these label properties. They just have the weight properties, so we're going to be using those. So I'll start by looking at Brodmann area 1 weight. So once again, this is a property that gives you the probability that every vertex is in Brodmann area 1, which, as a reminder, is part of primary sensory cortex. So I'm just going to turn this probability weight into an actual label where all the vertices that have a probability greater than 50% are considered inside the label. And then we're just going to go ahead and plot this. Now this uses a function called Cortex plot, which is part of NeuroPython. And this is a great function for making plots of 3D cortices, as we'll be seeing. You can ignore that warning, but as you can see, this looks kind of like a cortex, and we can see this bit of sensory cortex that's clearly labeled as part of Brodmann's area 1. Now, unfortunately, this is a hard plot to see, so I've included a few cells here that just kind of improve on the plot using different options of this function. I'm not going to go over these in great detail right now. I just want to show that this is all possible. Here we have it just kind of plotted in red. Here we're going to do the same thing, but we're going to make it a little bit transparent in order to make sure that we can see the curvature underneath the particular plot. And here we'll plot it on the inflated brain, and we're going to actually just plot the weight, the probabilities, to see what those look like. So that's kind of what the probability map for Brodmann area 1 looks like. And I wanted to show you one plot of property like thickness, which has a value that's kind of interesting everywhere. That looks kind of like this, and something we can see right away is that in sensory cortex, both here and back here in early visual cortex, you find kind of thinner gray matter, which is something that's been observed in many studies. Okay, I do now want to go over interpolation just very quickly. So this is a very common problem where you have data that you've processed on the surface and you want to put it into a volume for some kind of sharing or for some kind of comparison with some other volume data. What you need to do this is, first of all, a template image, and this just needs to be an image that's aligned with the brain in some way. Basically, you need to know what kind of image, what size, what resolution to make. So we use the subject's brain image as a template image. We're just going to pass in the header. There's some comments that kind of explain that here. And then what we want to do is we want to grab the thickness property from both the left and the right hemisphere, and we're just going to put that thickness property into the volume using linear interpolation. And this can all be done with a simple call to subject.cortex.image, which just takes your properties, your template, and the method you want, and we're going to have an output of floating point numbers. Now this may take a little bit to run for you guys. I ran it earlier, so some of the intermediate data is cached, and it will run a little faster. But once that's finished, we'll go ahead and make a plot. This again is that same kind of block of code to plot 3 slices of the images. And I'm just going to plot this using the hot color map from Matplotlib. And that will have to wait for this cell to finish. It'll take probably just about a minute. So let me see if there's some questions in the meantime. Someone asks, what is a slice of the image? What a slice of the image represents? Basically, since these are 3D images, usually in 2D images, we think of 2D images as having pixels organized into rows and columns. In a 3D image, you just have a single 2D image is one slice, and the next 2D image is the next slice. And so it's basically just the concept of rows and columns abstracted to 3D images as opposed to 2D images. And then someone asked what reflection means. So reflection would be like if we took a right hemisphere and we reflected it so that it looked like a left hemisphere. Basically, it makes it so that it's no longer, you can't rotate one hemisphere into a reflection, but you can absolutely make a reflection by just inverting all the x values, for example. Okay, and here we have the results. So it may be a little hard to tell that this is correct just by eyeing it, but let me draw your attention to a couple things. Down here, you can see some rather thick bits of Cortex that have very bright colors, and up here you can see some rather thin bits of Cortex that have relatively dark colors. So it looks like this pretty much worked. Now, again, I'm not going to go over the details of how this all works, but we can invert this process using the function image to Cortex. What I'm going to do is just to prove that all of this works pretty well, I'm going to produce versions of the 2 properties we just interpolated into the image by pulling them back out of the image, and they should be very similar to the original properties. So I'll go ahead and run this. This is much faster to go this way. That has to do with details of how the algorithm works. And then what I'm going to do is I'm going to plot each Cortex, both left and right. And what I'm going to plot is the square of the difference between the original property and the property that we just interpolated in and back out. So ideally, these should be very close to 0. And in fact, what we'll see is that they are very close to 0, although they're not exactly 0. And that's because, here they are. That's because there's a resolution limit in the images. So we get some instances where the interpolated value that comes back is just a little off. That's normal for this kind of process. Okay. At this point, let's go back to the slides. And we will talk a little bit about inter-subject comparison. So far, we've learned how to start with MRI measurements and how to determine where the various anatomical structures of it are located, and how to model and represent the cortex of a subject. And this is most of what you need to be able to make the animation I showed in the first slide. However, were we to try to morph the brain of one subject into that of another subject, we would have a problem, because the vertices of one subject don't actually correspond to that of another. So it's not clear at all how to deform the brain of a subject 1 to be more like the shape of subject 2. So in order to solve this problem, we need to use some methods for inter-subject comparison. So inter-subject comparison can be facilitated in both the volume and the surface, but the 2 are very different. Volume-based alignment is generally easier to understand, but it's also much more limited, so we'll just cover it briefly. In volumetric alignment, one typically treats the volume as a rigid body and only rotates scales and translates the brain. In essence, this involves finding an affine transformation matrix, or sometimes a limited affine transformation matrix with fewer parameters that, for example, only supports translation and rotation. And we want this affine transformation matrix that we find to minimize the difference between the voxel intensities of one image and that of another. And since there's at most 12 parameters in this kind of search, it tends to be pretty efficient. You can usually perform this pretty fast. This kind of alignment is most common for aligning bold and functional images together with structural images, and for motion correction. It's also used for aligning a subject's brain to a template or atlas brain, such as the MRI brain atlas. And in fact, the MRI brain atlas is the kind of de facto standard atlas. It's produced by McGill. Basically, aligning a subject with an MRI template brain will bring the anatomy of one subject roughly in line with that of another. So if you blend two subjects to an MRI template, they're roughly in line. But the alignment is not perfect since brains have very different shapes, as we hopefully gathered from that animation I showed in the very first slide. So there are many tools for these kinds of alignments, including BB Register, which comes with FreeSurfer. There's also FSL and ITK Snap, and there's a whole bunch of others. I won't talk about these anymore at this point, but you can find a lot of information about these online. So surface-based alignment is quite different. In comparison to volumetric alignment, it's a bit more sophisticated. Surface alignment typically starts with a similar approach as volume-based alignment. We need a surface atlas to which we can align the other brains. And to make this, we start with a bunch of brains from individual subjects who have already been volumetrically aligned, just like we do to make a volumetric atlas. So we then calculate the mean of all of them, and that produces an average brain that looks kind of like this. This is just a slice from an average brain. And this is actually quite very similar to what the MRI average brain atlas looks like. So notably, you can still see that there's kind of white matter and gray matter sort of showing up on this this particular image here. So we can actually still use this image to produce a model of the surface of this brain. And you'll notice that it's not nearly as wrinkled and folded as a normal brain, but that's actually good for alignment. And just something to note here that helps make all this make sense. Because the gray matter forms a thin sheet that covers all of the white matter, the gray matter surface of each hemisphere of the brain is topologically equivalent to the 2D surface of a sphere. And this is why we can create spherically inflated meshes, and just in general, spheres are generally easier to work with than highly folded triangle meshes. So they're important for surface-based alignment. And that should become obvious here. So shown here is on the right is the atlas right hemisphere, just like the one we just created, except in the last slide I just showed you a left hemisphere. So this is called the FS average hemisphere for FreeSurfer's average, and it was made from the group average MRI data that we just saw. And on the left is a white cortical surface of the subject that we've been looking at throughout this talk. To start with, we will inflate these into spheres. That looks kind of like this. Now, although this inflation greatly distorts the cortical surface geometry, it does attempt to preserve the distances between vertices on the cortical surface as much as possible. So we sometimes call the inflated sphere on the right the native sphere of the subject. Now we can look at these spheres and see some similarities, but their curvature patterns are obviously not the same, and we would like for them to be aligned. And although they look a little bit aligned, they're not exactly aligned. To emphasize this, I'm going to go ahead and highlight this sulcus in the atlas brain. This is actually the Cowpern sulcus, right near the occipital pole, where V1, or primary visual cortex, resides. If we just transfer the outline over to the subject's native spherical surface, we can see that it doesn't match up to something similar, though the sulcus right above it might match. In order to actually perform the surface-based alignment, we essentially perform a diffeomorphic warping of the subject's native cortical sphere, which treats it a bit like a sheet of rubber that can be stretched and compressed. And the goal is to warp the vertices such that the difference between the curvature of the two surfaces is minimized. And that looks something like this. So this is now the FS average aligned brain of that subject. And once we perform the alignment, we can see that the sulcal curvature pattern of the hemispheres line up much more closely. So quite clearly, not only does this Cowpern sulcus line up, but this sulcus here lines up here, and that's pretty clear for most of this subject. What this gives us is actually two sets of triangle meshes overlaid on top of each other. Now, due to the details of how these meshes are made, the FS average atlas mesh tends to be very regular, as kind of like the black triangles that are shown in this image on the left. The subject's mesh, shown in red, however, tends to have very irregular triangles, in no small part because it was just deformed to match another surface. If we want to use such a mesh overlay to convert data from one individual subject over to the average atlas, such as the FS average, and we might want to do that because we want to compare aligned data between subjects, we need to use interpolation again. So let's suppose we're trying to transfer data from the subject's mesh over to the FS average. no matter what kind of interpolation we're going to do, whether it's linear or nearest neighbor, we are going to be looking at an FS average atlas vertex, which is shown here in black, and it's contained inside this red subjects triangle mesh. So here I've just see that triangle enlarged. P is the FS average vertex, and the vertices A, B, and C are the subject's vertices. So first of all, nearest neighbor is just very simple. In nearest neighbor interpolation, we just use the nearest vertex to the FS average vertex, in this case vertex A, and we just copy the value of A over to P. So this is a very straightforward and often the ideal way to do interpolation, but it does have some downsides. For example, this vertex X is actually closest to the vertex D. So it's possible that there will be some break of continuity due to the fact that X is getting a value from outside of its triangle, the triangle that contains it. That kind of effect is usually minor though. But if you're worried about that, linear interpolation is a great way to solve that. So in linear interpolation, we do something very similar to in volumetric interpolation. We basically split the triangle into 3 subtriangles, and we use for vertex A, the weight of vertex A is the relative area of the opposite triangle A, and we just again do a dot product to get the value in P. In general, you'll encounter both volume-based and surface-based registrations in neuroscientific studies. They're different and have different advantages. That said, I would generally recommend surface-based methods in most cases that involve the cortical surface. Obviously, if you're studying cerebral cortex or brainstem or something, sorry, if you're studying the cerebellum or the brainstem or something, you can't necessarily use surface-based methods. But surface-based models are just more accurate in most cases that involve the cortex. As an example, if you take the ROI that's drawn in a volume space that's shown on the left, what I've done is I've drawn that ROI in the left, and I've transferred it to the exact same voxels in the subject that has been volumetrically aligned on the right. And if we zoom in on that ROI, what you can see is that in this subject, those voxels are all on one side of this sulcus here. But in this other subject, despite the volumetric alignment, those voxels show up on the other side of the sulcus. This is actually a very kind of error that occurs in volumetric alignment. And surface-based alignment is just not nearly as vulnerable to this kind of error. And in fact, this has been very well studied over the years. FreeSurfer was actually, the first publications about it came from 1991. And since then, people have studied this many times. These are just, at the bottom of this slide are just a few of the example papers that have concluded that surface-based anatomical alignment is superior for inter-subject comparison than volumetric alignment in most cases. In particular, I want to point out that this first paper, Rosenka et al. 2018, not only compared volumetric and surface-based alignment, but also compared a form of diffeomorphic volume alignment that I haven't talked about in this talk, and found that even it was not as good as surface-based alignment. So in general, surface-based alignment is a great way to compare between subjects if your data are on the cortical surface. Okay, so at this point, let's go back to our notebooks for the last section on inter-subject surface alignment. Okay, so before diving too deep into this, I wanted to just plot a few of those spherical hemispheres. So what I'm going to do first is grab the FS average subject, which is that atlas subject from FreeSurfer. We grab this the same way we grab any subject. It's just FS average is just a special FreeSurfer subject, basically. I'm going to grab the hemisphere we're using, left hemisphere for me for the FS average, and the subject's left hemisphere as well. I'm then going to get the subject's native sphere, which I do in exactly the same way that I get a surface, except instead of using surfaces, we use what's called registrations. Registrations just refers to the spherical inflated hemispheres that are designed for inter-subject comparison. So basically, we're going to grab the native registration or native spherical surface of this subject. We're going to grab the subject's FS average aligned spherical surface, and we're going to grab the FS average subject's native surface. So basically, this is the original native sphere. This is FS average's sphere, and this is the subject's native sphere after alignment to the FS average. So that we can plot them all in one scene, I'm going to give a slight, I'm going to just slightly translate 2 of them to be either over to the left or over to the right with this translate function. And then I'm going to make an IPy volume figure. I'm going to plot each of these three spherical surfaces, and I'm going to show the figure. So that should take just a second to plot. And what we should get are three spherical hemispheres all kind of in a row in the figure. Yep, and here we are. So these are all kind of plotted together so you can kind of look and compare. What you might notice is that these two, so here on the very left is the subject's native sphere. This is the FS average aligned sphere. So this is after this sphere has been aligned to the sphere on the right. The sphere on the right is the FS average sphere. So what you can probably notice is that although these two are very similar, this middle sphere is a little bit more similar to the one on the right. And that's, I think, easiest to see by comparing, for example, where these kind of lines of curvature, which correspond to the corpus callosum, fall in the three hemispheres. So those are the actual spheres themselves, but most of the details about dealing with them are hidden from you if you do interpolation through NeuroPython. So let's just, as an example, interpolate thickness from our subject over to the FS average. This is something we might do if we want to, for example, compare the thickness on a particular location of cortex between a bunch of different subjects. So we'll go ahead and grab the thickness property. We're going to use linear interpolation. And the interpolation itself is done just with this line here. So we're going to interpolate from our subject's hemisphere onto the FS average hemisphere. We're going to interpolate that property that we just grabbed. We could also just use the name thickness here, I suppose. And then I'm just going to make a plot of the FS average hemisphere, the inflated surface in particular, and I'm going to color it according to this property that we just interpolated. So this should run pretty quickly. And there we have it. So here we have our subject's thickness data plotted on the FS average hemisphere. Now, notice here, this is slightly different shape than the plot we made earlier. If we just scroll back up and find that plot, you can see that the shape here is quite a bit different. That's because this is the subject's inflated hemisphere instead of the FS average inflated hemisphere. Also, it looks like I used a minimum value of 1 here, so this looks a little darker, whereas down here, I think I used a minimum value of 0. Oops. Yeah. So that's why the colors are a little different. If we just rerun that, they should look more similar. And then this last cell is the last cell in our demo today. And in it, we're just going to do exactly the opposite thing. So we're going to interpolate from the FS average. Oh, here we go. Yeah, this looks a little more similar. All right. So in this last cell, we're just going to interpolate from the FS average onto our subject. And as an example of why you might want to do this, it's much less common, but you might want to plot the FS average curvature on your subject as an underlay. So what we're going to do is we're going to take the FS average hemisphere, we're going to interpolate onto the subject hemisphere, and we're going to interpolate the curvature data. And then we're just going to, basically this will make a copy of our subject hemisphere with the new curvature data overwriting the old one. And then we'll just make a plot using that new hemisphere make, and we'll see that we get the FS average curvature on our subject's inflated mesh. And there we have it. This is clearly a different shape than our FS average mesh that you can see here. So this is our subject's mesh, but this has the FS average curvature plotted on it. Okay, that concludes the presentation part of my talk. Here are a number of resources, and as a reminder, you can run this tutorial notebook we've been looking at. You can run it all in a Docker image. There's instructions for how to do that at this this GitHub repository, as well as in the curriculum directory that you will have. Thanks for your attention, and I'd be happy to take any questions.
So there are a few questions here in the Q&A that I'm not sure you've gotten to yet.
Okay.
One that was asked and is sort of interesting, and now is also in Slack, is what's the difference between a template and an atlas?
I would say that the answer to that is that we neuroscientists have historically been a little sloppy with our language around the two. and they're often used a bit interchangeably. I think Atlas is generally used either to refer to for example, the FS average subject itself is kind of called an Atlas subject because it's something that you align brains to. At the same time, we also sometimes say things like there's an atlas of activation, functional activity that's based on the average of a bunch of subjects, for example. And so in a sense, atlas is used just to refer to the fact that something is kind of a measurement related to lots of subjects. And I'd be interested if you have a different opinion about that area or anyone else listening, but that's my sense of how it's generally used.
Yeah, my impression is also like yours, that it's not necessarily that distinctive. I think of Atlas as something that tells you where different things are, whereas a template is sort of like an image of the entire thing, right? That you use, for example, for illustration. So the MNI brain, I think of that as a template, where I think of the AAL atlas that tells you where different parcels are as in an atlas. But yeah, the distinction is kind of tricky. And we have a Slack thread for that, so we can continue the discussion there. Somebody else asks, let me find this. There was some question about the differences between neuropathy and FreeSurfer in terms of the interpolation methods and so on.
Okay, so FreeSurfer, I probably should have included this somewhere, but, and I'm going to preface this by saying that I haven't used the most recent version of FreeSurfer yet, so I'm not certain this applies to the most recent version, but in some of the older versions of FreeSurfer, there were tools included called MRI Vol to Surf and MRI Surf to Vol, which would ostensibly do this job of transforming data from the volume to the surface and the surface back to the volume. They used an algorithm for those particular transformations that were based on starting at, for example, if you want to convert data from the surface into the volume, what they would do is they would start with the position of the white matter vertex, and then they would draw sort of a vector up to the pale matter vertex through the gray matter. And they would populate voxels at different distances that landed at different distances along that vector with the value for that particular vertex. That is a fine, in theory, way to go about it, but the problem is that a lot of voxels in the gray matter don't end up on those vectors. There's actually a little space, and additionally, there are a lot of voxels that end up on multiple vectors, and it gets very messy trying to figure out a very simple interpolation in that particular way. And so what ended up happening was, if you used Vol to Surf or Surf to Vol in FreeSurfer, you would end up with very patchy data where you had big holes in the interpolated result. And again, I don't know that this applies to the most recent versions of FreeSurfer, but the versions of interpolation used by NeuroPython will fill in the entire gray matter, and additionally have tools for doing things like pulling out a particular layer. If you're interpolating onto the surface, you can pull out a particular layer, like just along the mid-gray or just along a quarter of the way between the white and the pale. In general, the NeuroPython tools are a little bit more flexible, at least as of older versions of FreeSurfer.
I'll do one more, and then I think we'll break, because we can take a little break before the next thing. And I'll sort of combine two. There's a question here about specific templates for children or adolescents, and also another question about how do we think about abnormal brains when we work with brains that have damage or abnormalities. How do we do that? Yeah,
these are both really good questions. And actually, Ariel, you may know the answer about the children's brains better than I do. I'm not sure, but funny fact, Ariel and I actually met because we were originally working on a project together that dealt with children's brains. This was ages ago, but and how to deal with them in these kinds of contexts. And my recollection is that we didn't actually solve that problem. To my knowledge, people are still collecting data to try to answer this question about how well children's brains and adult brains align. I think there are a few groups that have made, they basically made their own version of FS average using children's brains, but I don't offhand know of any that are public, but that may just be because I don't personally study children. So I think this is probably a question that would be better asked in Slack, and there's a good chance there is some template out there. Now the problem with brains with like with a lesion, for example, if you have a subject who is missing a big piece of cortex, that becomes a much bigger problem. And the reason for that is because when you do that alignment step, the surface-based alignment step, where you deform the spherical brain to match the FS average sphere, that is a global minimization that attempts to minimize the curvature of the brain simultaneously, the difference in curvature of every point on the brain simultaneously. So if you have a big chunk missing, there's nothing to really, the algorithm is going to try to fill that space in with stuff anyway. And so what you often get is a very strange kind of alignment between subjects. And I guess I would say that I wouldn't recommend using FreeSurfer's straight up inter-subject alignment for a subject with a big lesion. This goes even so far as if you have a lesion, so if you can see my mouse right now, I've looked at subjects that had lesions up in this kind of area of the brain, closer to frontal cortex. And even looking at the occipital pole, you'll still find problems with the alignment back here because it's a global minimization. So in general, you have to be pretty careful with subjects that have any kind of damage or any kind of missing cortex with these alignments. I would just recommend checking them carefully by hand and making sure they look okay if you're going to use them.
Okay, great. So with that, I'll suggest that the remaining questions go to Slack or to Neurostars following this week. And thank you so much, Noah, for the presentation.
Yeah, thanks everyone for coming.
Take care.